It's time for the 1430 Connection on 1430 WNAV and 99.9 FM, spotlighting news, newsmakers, and important community issues. Now, with this week's edition of the 1430 Connection, here is WNAV news anchor Donna Cole.
Welcome to the 1430 Connection. I'm Donna Cole. Today I have the pleasure of talking with Dr. Sarah Donelan. She's a postdoc at the Smithsonian Environmental Research Center in Edgewater, and she discovered something pretty cool. So that's why we're talking to her today. Sarah, thank you so much for joining me.
Thank you so much. I'm happy to be here.
Are you continuing at Smithsonian Environmental Research Center now?
Yes, I am still at Smithsonian, and I'm finishing up my work on oysters and now transitioning to work on invasive species. So there's a lot of great work happening at SERC, and I'm happy to be a part of it.
And what was your PhD in, if you don't mind me asking?
I worked on how parental exposure to environmental stress affects animals, which is similar to the work that I'm going to be telling you about today in oysters. So I worked with snails and crabs up in New England.
How did you come to develop this fondness for crustaceans?
Well, I grew up on the rocky intertidal shores of New England, and there's a lot of fantastic, really interesting marine invertebrates that live there, like snails, like crabs, even things like oysters that appear kind of not to do much but are actually really interesting. So there's this whole kind of secret life of these invertebrate marine creatures that I developed a fascination for early on.
Okay, is this fascination just into research, or do you enjoy eating them too?
Oh, I certainly enjoy eating them. I think one of the benefits of working on oysters in this experiment was getting to see all the different oyster farms that are really popular around the Chesapeake Bay, and of course purchase some oysters myself.
Right, and we're going to talk about where you purchased the oysters, because that was in Cambridge at another facility that we should know about. Let's talk about your study, and I will say in the email that I was sent that introduced you and the study, it says Sarah Donelan, one of our postdocs, discovered that warming waters and low oxygen, two stressors that often coexist in the Chesapeake Bay, can trigger young oysters to focus more on growing their shells and less on the meaty tissue inside. This could pose a problem for the seafood industry, since oyster farmers rely on shell size as an indicator of how much tissue is inside. It could also create an issue for the restaurant industry, everyone involved with oysters.
Yeah, I think, the management implications of our results are still not clear, but what we found is that kind of just like humans is how I like to think about it, oysters are affected by things that happened to them when they were young. So what I did in this experiment, and I also conducted this experiment with two other scientists at Smithsonian, Denise Breitberg and Matt Ogburn. We took pretty young oysters, what we call juvenile oysters, and we put them in this stress exposure experiment. So some oysters were exposed to one stressor, like high temperatures or low dissolved oxygen, and some oysters were exposed to both. So low dissolved oxygen is a thing that happens in the Chesapeake Bay every summer, but can also happen over a daily cycle in rivers and tributaries of the Chesapeake, like the Choptank River. And low dissolved oxygen is a problem for oysters because they need oxygen in the water in order to breathe. And so if there's not a lot of oxygen available, that makes it harder for oysters to breathe.
Let's start with how you got the oysters.
Definitely. So there is a humongous, fantastic oyster hatchery at the Horn Point Labs run by University of Maryland in Cambridge. And they were absolutely wonderful to work with and produced the oysters that I used in my experiment. And they produced a lot of the oysters that are sold or given to oyster farmers in Maryland and that end up on your plate. Now,
what size oysters? Are we talking about spat, or were they a little bit more grown?
They're still considered spat, yes. They were about a centimeter big, so a half an inch about when I started working with them. And they were about 4 months old. And so those spat oysters were exposed to these different stressors of the high temperature and the low oxygen, or some were exposed to both of those. Then after they were exposed for a really brief period of time, only 18 days, I took the oysters out of the stressors and just let them chill out for a while. And about 60 days later, I re-exposed the oysters to those same stressor categories. And what we found is that the early exposure to stress affected how oysters respond to later stress. Oysters that were exposed to this double whammy of stress, both as spat and, 60 days later, were less big. They grew less tissue compared to their shell than all of the other oysters. The reason that matters is because, when you are eating an oyster, which many of us like to do, what you really are interested in is eating the tissue. You're not eating the shell. And so you want to have as much meat per shell as possible. And my results suggest that there could be these long-term effects of stress on oysters that affect their tissue growth. It wasn't like a humongous difference in growth. It's not like you'd open up an oyster and it would be a teeny tiny, tissue, meat amount available inside. But it's something to consider moving forward that, we need to think about the long-term effects of stress on these oysters.
Oysters are one of the best filtration systems anywhere, but especially in the Chesapeake Bay. That's what we're talking about. And they produce the oxygen that not only they need, but all living life in the Chesapeake needs, yes?
They're really important filter feeders. I'm sure you know this, but a really fun fact is that one oyster can filter 50 gallons of water in a day, which is about the volume of a bathtub. And so there's, oyster reefs have thousands of oysters on them. So they're extremely important filter feeders in the Chesapeake. And so it's important for keeping nutrient levels down, like nitrogen levels in the bay down. That can affect oxygen levels in the bay. So it's a little more complicated, but they're important parts of the Chesapeake Bay ecosystem that help maintain a healthy balance.
If there's less meat by thousands, millions of oysters in the bay, that too would create an issue for the oxygen levels, would it not?
Yeah, no, it definitely could. I think another interesting thing about less meat is that oysters, depending on the amount of meat, they are either male or female. So oysters with more meat are generally female when they're bigger. And so this could have effects on when oysters change from male to female, and then, how oysters reproduce and how populations grow throughout the Chesapeake Bay.
We're getting into sex talk. Who knew? I actually did not know that about females. It's all of my time on the Chesapeake Bay and loving oysters. I had no idea the more meaty are often females.
Yes, they're fascinating organisms.
I've learned something new today. We need to take a quick break, so we will be right back on the 1430 Connection. Welcome back to the 1430 Connection. I'm Donna Cole, talking today to Dr. Sarah Donelan. She's a postdoc at the Smithsonian Environmental Research Center in Edgewater, otherwise known as SERC, and we're learning today about a study that she and two others did at SERC that seems to show that when exposed to stress, such as low oxygen, higher temperatures, oysters don't develop as much meat, but their shells are thicker, yes?
Yes, that's right.
So this started in 2018. It is now 2021. I know that these studies, and I've seen the study, and it's huge, and it's extremely informative, and there's a lot there. How does this happen that it goes from 2018, most of the studies were done in 2018, that it doesn't get published until 2021?
Yeah, that's a great question. So the experiment finished in late 2018, and the experiment had about 1,200 oysters. And what we did is we tracked the individual growth of those 1,200 oysters. And so, as you might imagine, that's a lot of data, that's a lot of oysters. And so the first step is just kind of figuring out what's going on, looking at those data and seeing if we found anything interesting. So that can take a decent chunk of time. For example, I was still working on the data in 2019 when I ran other experiments with oysters that I'm now writing up. So it's a long process. Once you work up the data, you start writing a paper that communicates your results, and that can take some time once you have submitted your paper to a journal, which is a typical process in research. It can take some time for it to get through the whole publication process. It indeed does take a long time, and I'm glad the results are out. And it comes at a fortuitous time because it's Oyster Fest in Annapolis right now. So
exactly right.
It's a great time to be thinking about oysters or talking about oysters because hopefully people are eating them. I think it would be fun for people to think about the early life of the oyster that they're eating right before they eat it and how, the environmental conditions that it experienced are affecting what they're eating today.
And these two large stressors, higher temperature, lower oxygen, all of this has been human-caused. The question is, can humans turn this around? With regard to the oysters, what do we do? How do we save them?
What our results point out, it initially doesn't sound good. It sounds like, okay, double whammy, oysters grow less tissue. That's bad. But actually, we can harness this information to help our oysters. So farmers and scientists kind of already knew that juvenile and baby animals are really vulnerable to stress. And our results corroborate that information and suggest that if we can limit the amount of stress that young animals are exposed to, animals like oysters, then we can help them grow better later and reproduce better later in life. So this just might mean that we need to pay a special careful attention to where oysters are being grown, and it might impact how they grow later.
More focus on aquaculture?
Oh, aquaculture aquaculture is such a great economic and ecologically important mechanism in the bay for helping restore oysters. The watermen or farmers who grow oysters have such a good knowledge of their natural history and knowledge of the place where their farm is located. And so it will be good information for them to be thinking about where their oysters are grown, which I'm sure that they already are, but just, more information about where it could be good to grow their oysters, areas that are high in dissolved oxygen and that have normal temperatures.
Let's take another quick break. We'll be right back on the 1430 Connection. Welcome back to the 1430 Connection. I'm Donna Cole, talking today to Dr. Sarah Donelan. She's a postdoc at the Smithsonian Environmental Research Center, otherwise known as SERC, right here in Edgewater. Sarah, what does this mean for lobsters? What does this mean for crabs?
Yeah, great questions. I think that similar effects could be happening in other, commercially important marine invertebrates like lobsters and crabs that we also love to eat. I know that there's some ongoing work occurring on those kinds of species, looking at how exposure during the egg to different stressful conditions affects them later in life. So I think the jury is currently still out, but we see these types of early life experiences affecting animals across the animal kingdom, from oysters to bugs to fish, and also operating in, plants. And so this phenomenon is occurring across the animal and plant kingdoms, and so I have no reason to believe that it's not happening in other commercially important species. And so it's, these early life effects are something that we need to keep an increasing eye on, both as scientists and as lovers of seafood from an industry perspective.
What's coming next is perhaps the most odd question ever asked about oysters, but keep in mind that I am a helicopter mother. At what point do they separate from their mothers and aren't stressed by that separation?
Yeah, no, great question. The male and female oysters release the sperm and the egg, more sex talk, release sperm and egg into the water, and then the egg fertilization happens in the water. So oysters are never with their parents, pretty much. That fertilized egg develops in the water for about 2 weeks, and then once it's reached a certain stage, is ready to settle back down onto some hard substrate like a dock or like an oyster reef. So it's possible that those larval oysters might settle on the same reef as their parents, but the parents aren't, caring for the oysters or anything. So it's really how that juvenile baby oyster by itself experiences the environment that appears critical here.
I should also ask you about the room of doom, because it sounds bad, and this is a room at SERC, apparently.
Yes, that's right. So decided to come up with a fun name for it. It was named before I arrived, but I do enjoy it. So it's technically an acronym. DOOM, it stands for Dissolved Oxygen Oyster Mortality, but it's a very apt name for the room because there's lots of tubes and gauges and gas bubblers everywhere around the room. So it's a very stressful room to be in for not only the oysters but also for the scientists. So SERC has really great experimental facilities for species like oysters, which is why there's such great work on coastal systems coming out of SERC.
I was just about to say, with your background coming from New England and studying, what did you study in undergrad years?
In undergrad years, I worked on streams.
Okay, so with this background, arriving at SERC must have been like a dream come true.
The facilities at SERC and the scientists at SERC are really, top-notch. I feel that the community at SERC does a really great job translating, what we do on the basic science level into information that Marylanders care about and that hopefully managers will care about. So it's a really exciting place to be practicing science, yes.
So how do you get, other than through media, which I think many of us shared this information, and this show will obviously be shared, how do you get this information that might be that is so critical to the seafood industry to those in the industry? We
have attended the Waterman's Expo, different shellfish meetings that were supposed to happen prior to COVID. And, I want to stress that I think there's a lot more work that needs to be done before I make specific recommendations to farmers or managers beyond that. We need to think about these early life stages for oysters and how, stress exposure might affect them later. But we're still in the early stages of this work. Okay, so you're not ready yet for the film on stressed out baby oysters. Not quite yet, but I do look forward to that day.
So what is next for you? How does this? What will you be doing next to further the study, and/or what is your next study?
These oysters had siblings that I kind of held for a longer period of time in no stress conditions for a whole year. And then I exposed those siblings to stressors. So that's what I'm working on right now, is kind of the even longer term effects of this early life exposure. And what I'm finding is that these early life exposure to stress can persist even for a year in an oyster, which is pretty remarkable. And there's just so much more work that needs to be done. And hopefully I'll be able to do some experiments, some further experiments to look at it.
Well, thank you for taking the time today to spend some time with me and the listeners.
Thank you, Donna. It was a pleasure.
For more information on SERC, you can visit serc.si.edu. I will be sharing the study link in the YouTube and Facebook version of this interview. This is Donna on the 1430 Connection. We will see you next week.